Welcome to this next episode in our EMIS at Home series, a program developed by EMIS to share thought leadership safely and securely from their homes during a pandemic. We are pleased to welcome Supriya Menezes, who will be moderating a panel discussion on the challenges of managing rare disease studies in ophthalmology.
I am Supriya Menezes, Director and Biostatistician of the Ophthalmic Therapeutic and Research Unit at EMIS. I'm joined today by a panel of experts in the world of ophthalmology research. With over 20 years of experience, Dr. Tracy Clemens is Chief Research Officer for EMIS. She currently oversees 8 therapeutic and research units, serves as the principal investigator and lead biostatistician for a large global ophthalmic pharmaceutical program that is currently conducting 4 phase 3 clinical trials. Also as the lead statistician for the MACTEL Project Data Coordinating Center, sponsored by the Lowy Foundation, and she served formerly as the principal investigator on the age-related eye disease studies, ERIDS-2, among others. Dr. Brian Brooks is the clinical director of the National Eye Institute and chief of NEI's Ophthalmic Genetics and Visual Function branch. His research is focused on the causes and mechanisms of inherited eye diseases, particularly those that affect children, such as uveal coloboma and albinism. He has authored more than 70 peer-reviewed manuscripts and 20 review articles on the genetics of eye diseases and has received numerous awards for his work. And Dr. Barbara Blody joins us as the medical director of the Fundus Photograph Reading Center and professor in the Department of Ophthalmology and Visual Sciences at the University of Wisconsin School of Medicine and Public Health. She is also a medical retina specialist. Dr. Blody's research interest centers around clinical trials for macular degeneration, diabetic retinopathy, posterior uveitis, and other retinal diseases. Thank you, everyone, for joining us today. I would like to move on to, I think, what everyone is interested in, and that is the key challenges in running ophthalmic rare disease trials. Dr. Brooks, what are the challenges in defining and agreeing upon classification of rare diseases?
There are multiple challenges that come with running clinical studies, clinical trials for patients with rare disease. Perhaps the first and most obvious is that these conditions are rare, and so finding the patients that will fit your inclusion and exclusion criteria in your trial is a less than trivial matter that maybe we can discuss in a little bit. The other thing that comes to mind is that you have to define a suitable outcome measure, right, for rare disease, just like for common disease. In many cases, we have a lack of clear natural history for a given disease, and so we need to learn that, perhaps, before we can conduct a rational clinical trial. In many cases, for instance, of degenerative diseases, visual acuity, which would be a classic go-to in ophthalmology, may not be your best outcome measure to look at in the first place. And also within diseases such as retinal degenerations, a rare disease, there can be very slow progression of disease. And so if you're designing a trial that you would like to complete in a modicum of time, you have to be able to follow something that can be captured within a couple of years' time. One additional thing is that a lot of rare diseases affect kids. And so whatever your outcome variable is would have to be something that the kids could cooperate with, for instance. And so that makes more advanced imaging, for instance, challenging on children. And sometimes by the time they can cooperate, the disease has already progressed to a point where you wouldn't be able to follow the things that you would normally want to follow. So an example of that might be retinitis pigmentosa 2, which is an X-linked form of retinitis pigmentosa, which in the affected males is extremely aggressive in its progression.
It's it's really important to underscore the need for consensus among investigators. First of all, simply on those definitions that Dr. Brooks was mentioning. So the investigators, it may take some time to get aligned in terms of what this disease is, what are the stages of the disease, and what do we know about the natural history. So sometimes it takes not a treatment trial, but a natural history trial first. That's often a very good approach so that the investigators can be aligned. And I think that that happened in diabetic retinopathy 40 years ago. There really wasn't a common language. There wasn't a common way forward until a number of meetings, which did lead to the diabetic retinopathy study. So it's the same for newly described diseases or diseases where we now have the possibility of treatment. And now we do need to step in and get those definitions and get the investigators to think in a pretty open-minded way, because that's the other, I think, important challenge is investigators, we all have our biases, how we think a patient should be treated or followed. And we really need to listen to each other to find out how to go forward.
Just on the statistical end, one of the big issues in conducting trials in rare diseases is that small sample sizes are associated with studies of rare diseases and can restrict the study design options, also replication, and also the use of inferential statistics, which means that novel and innovative statistical designs may need to be considered in order to assist in assessing the evidence of efficacy and safety.
If I may maybe jump in, maybe I can give a real world example of the challenges that are faced by researchers. And to build on Barbara's point, sometimes you need to think out of the box and you need to create your own novel outcome measures. So a classic example in the ophthalmology rare disease space is the work that was done through Spark Therapeutics for Luxturna, the gene therapy for RPE65-mediated Leber congenital amaurosis. Now, this is a very aggressive form of retinal degeneration. These children are born with decreased acuity, and it progresses pretty rapidly. So many of the problems that we outlined just now were encountered in this patient population. So the investigators created a novel outcome measure, which was the so-called low luminance maze test, or LLMT. What the test involved was having the child walk through one of a series of mazes where there were specific obstacles or things you had to step over or step down a step, for instance. And they would do this test about how long it would take for someone to wind his or her way through the maze under different luminance conditions, so under relatively bright light and under dim light, and compare that with controls. And in fact, that novel outcome variable was agreed to by the FDA and was one of the reasons why it was approved as the first gene therapy for an ocular condition a couple years ago now.
Dr. Blody, could I ask you a little bit more if you could elaborate on maybe diagnostic tools and multimodal imaging that needs to be kind of the challenges associated with that in designing these trials, or from your experience with Stargardt's?
I'm fascinated by sort of the imaging possibilities we have. I would say at times we need to be cautious that we don't do too many tests. So in clinical trials, especially with children involved, we can't have a 3-hour visit. The patient, the young person, is not going to tolerate that, and it's frankly too tiring. So I think we need to be very cautious about the number of tests that happen, imaging tests at any one visit. But I will say for children, they've done really well with OCT and OCTA. That's an easy one. I've actually seen children do really well with ultra wide field imaging. It's not as bright as a standard fundus camera, and it's not as many flashes of light. So those two are really, I think, very easy, generally easy. Certainly the OCT and OCTA could be done at every visit. But there are definitely challenges with other tests. So a fluorescein angiogram in a child or a young child may require an evaluation under anesthesia. The imaging tests, I think, again, we've seen now in trials, we rely on certain standard tests that may occur at each visit. OCT is a common one, but then only use the other tests at milestone visits. So if a photograph or an angiogram or autofluorescence is needed, that may be done at 6 months or 1 year. Some of these trials are long, they need to be long. So a yearly visit with more imaging is worthwhile, but I wouldn't do that at every visit. That's too much.
Dr. Brooks, in ABCA4 retinopathy studies or any other ocular pediatric studies, can you share with us any challenges related to maybe the duration of the study or disease progression that kind of needs to be taken into consideration?
Yes, so many of these conditions are slowly progressive. And so trying to find an outcome variable that will change significantly over a modicum of time is a bit of a challenge. As we previously mentioned, having good natural history data across a variety of modalities is the first step in trying to define what it is you're going to follow. And in an example of ABCA4 retinopathy that we have here at the National Eye Institute, we have been following a cohort of patients with ABCA4 retinopathy under a natural history study now for several years. And we've gotten approval by the FDA for repurposing a small molecule, metformin, which is used in the treatment of diabetes, as a possible treatment for ABCA4 retinopathy. And here, because we're looking at a rare disease, doing a phase 2A study that was a randomized placebo control would have a sample size that would probably be prohibitively either long and or large for doing the initial evaluation. So in discussions with the FDA, we have been able to define, for instance, that the rate of change for an individual patient using a specific OCT measure is linear when you either do a log or a square root transformation. Now, one of the things that we run into is that even in a genetically comparatively homogeneous group of patients, so all of these patients have 2 mutations in the ABCA4 gene. You might offhand think that they would all be remarkably similar, and in some ways they are. But each patient progresses kind of at his or her own rate, yet they seem to follow their own course, their own line. So how does that help us with trial design? Well, in discussions with FDA, we've been able to design an open label study, in this case, using the existing carefully and systematically collected natural history data as our baseline, and then to determine whether when we intervene, in this case with oral metformin, do we make the slope of that progression shallower? Do we have an inflection point? And with that, we think that we will be able to detect a change using this imaging OCT outcome measure in about 2 years. So it's still long for a typical trial, but is tractable, is doable because of the trial design and because of the natural history data that preceded it.
Supriya, if I could jump in right there too, I just want to comment on how I think that is such a great design. It truly represents personalized medicine. I think every patient would be grateful for knowing that their own path is being monitored and the trial is following them based on their rate of degeneration. And I'll also just go back to this topic of the FDA. I think one thing that we need to recognize as investigators and I think the public as well, is that the FDA does assist investigators in clinical research at an early stage. Even if it's not a drug trial yet, they will be there to provide guidance and advice. Certainly by the time it is a drug trial, the FDA needs to be involved, but I generally see that in a very positive light. I think that they're providing good feedback so that the trial design is going to hold up. You don't want to be at the end of a trial and then be told this is the wrong design or you've chosen the wrong outcome. So I think involvement of the FDA is something that really has improved clinical research in our country over the last few decades.
Speaking on trial design, Dr. Clemens, it's always crucial and an important component. Could you share some examples of alternative study designs that may be appropriate for a rare disease trial?
Certainly, Supriya. One example is called a blind start approach or blind start design. In this type of design, all patients receive the minimum duration of active treatment. So for example, let's say that you want to treat participants for 2 years. So all participants will receive the IP for 2 years. However, the participants are randomly assigned to begin the active treatment at different predefined time points. So they may be randomized following, say, 8 months, 16 months, or 2 years of, say, a sham or placebo treatment prior to being assigned the active treatment. This study design offers an estimated treatment effect size that's similar to that of a randomized parallel group study. In its power to detect a real effect on clinical outcomes that is very similar to that of the traditional parallel study design with the same sample size. Other types of designs or trials that are being used are called umbrella trials, where you test many therapies for one rare disease. Also basket trials, where there's testing of one therapy against many rare diseases. And then platform trials, which are like umbrella trials, but you use a decision algorithm to let therapies enter and exit your platform.
Dr. Blody, what has been your experience using a biomarker as a primary outcome instead of visual acuity, for example?
Yeah, thank you, Supriya. That's such a good question, especially in ophthalmology and especially in retina. And we've had imaging as part of retina for many decades. And so a primary outcome may be based on imaging results, and maybe the oldest example I can think of is diabetic retinopathy. So the change in someone's diabetic retinopathy status is not dependent on their vision. It's dependent on what we see in the retina and actually by the photographs that are taken. So a patient will get color fundus photographs a specific way to map out the retina in order for the investigator or reading center to determine what level of retinopathy is there. Their vision may be perfect, but they have severe proliferative diabetic retinopathy. So that's an imaging biomarker. There are now some other examples. For a while, that was the only imaging biomarker that the FDA would accept. So that was kind of an exception. But we do see other imaging biomarkers that are now accepted by the FDA as a primary outcome. And one is area of atrophy, when we're looking at trials where there's atrophy, geographic atrophy in AMD, Stargardt's disease, other conditions where it's atrophy. That's really what we're looking at changing with medication. Can we slow down the atrophy or even reverse the atrophy? The other pretty novel imaging is looking at the OCT, and Brian mentioned that as well. But in one condition that affects older adults, middle-aged, to older adults is called macular telangiectasis, or MACTEL. And their ongoing trials are using the OCT as the primary outcome. And it's not just the OCT scan, it's one layer of the OCT scan, which represents our photoreceptors, our rods and cones. So that's quite a big jump, I think, for the FDA to make and allow these imaging biomarkers to be used as a primary outcome. And if I could make a comment about that, it does really set the bar high on quality of imaging. So if you're in a site where the quality of imaging is poor, you could be in trouble, because that is the outcome for that patient. A reading center, and there are a number of excellent reading centers in this country and outside of the US, that really we all work hard to make sure there's quality imaging at the site, so that the photographers or technicians are trained in a specific protocol and how to get a good quality image. And we also certify the equipment, the imaging system, the OCT system or the camera system, to make sure that the images are the best possible images. You don't want to have an image that's ungradable, because now you've lost the chance to get that primary outcome.
Thank you. That's definitely a point we've noted, especially when we work as the data coordinating and statistical center. The quality of the data is so crucial, and then the grading that comes from it, given that some of that is primary outcome. So Dr. Clements, can you share suggestions on how to demonstrate statistically significant results when working in a rare disease trial that could have lower power due to a small sample size or maybe a small treatment effect?
Certainly, Supriya, thank you very much. You may want to consider, especially if you have multiple primary endpoints, to conduct global tests for multiple endpoints. For example, rank sum tests based on the sum of ranks of data from 2 endpoints from each patient, or also combined test statistics based on 2 test statistics for treatment comparisons for each endpoint. Also, this goes into, especially MS's experience as a CRO, there are some quality assurance measurements, also training measures that should be taken into consideration to reduce noise and variability of your outcome measurements and potentially increase your statistical power. You should always include a manual of procedures that has standardized methods and procedures for your outcome assessments, and train the sites on these protocols. And Dr. Blody, that also gets into the imaging and the protocols for collecting those images and ensuring that the photographers are following those protocols. Also, work with the sites on the importance of minimizing dropouts and missing data. And lastly, ensuring that we're training personnel to encourage our participants to complete the study, even if they stop study treatments early.
For everyone, I just kind of want to piggyback on one thing you said, Dr. Clements, for Dr. Blody, and that's about engaging a reading center early on a study. Dr. Blody, can you comment a little about that and how important it is?
The need for a reading center is there when you have, generally, when you have multi-center trials. So there's certainly studies that are done at one center, and the PI or other researchers can do the work of analyzing the images. But when we're looking to standardize across sites, and now we're seeing clinical trials with hundreds and hundreds of sites because it's global trial, we really need a standardized approach. And so having the reading center, being able to spend some time with the investigators virtually or in person, to share what we do and why we do it is really important. So at an investigator meeting, I go to a lot of investigator meetings to really get that point across, that we're here to help the sites and we're here to help things be standardized. And that's just important in order to get, as Tracy mentioned, that good quality data. So getting a reading center on board can really get those investigators and the imagers, the photographers or technicians, trained and thinking about the quality of the images. I'll just put a shout out to the need for the coordinating center. So I think as we hear from Tracy, ophthalmologists will all say, well, I need a statistician, and that's true. But you do, beyond the statistician, who's going to be important throughout the trial, I really believe in coordinating centers because of the ability to reach out to the sites and to bring sites together. They're the glue that will keep sites answer questions, help sites keep doing things in a standardized fashion, troubleshoot issues they may have throughout the trial. That would be hard for a principal investigator to do him or herself, even if there's a group of principal investigators. It does take the work of a coordinating center to see that that trial is going up and started, and then able to kind of maintain good quality data and the support. We really want those sites to feel involved and to have a voice.
That kind of leads into my next topic of discussion, and that is some of the challenges that studies face with recruitment. Dr. Brooks, what are some of the approaches that work well to recruit patients in rare diseases?
So recruitment for any clinical trial is of utmost importance. And we've already talked about some of the hurdles that you can face when you're trying to design a trial, carry out a trial for a rare disease. I think in terms, it's probably true of all clinical trials, but it's especially prominent in rare disease that the recruitment and the continuation of a study, the maintenance of a patient in either the placebo or the treatment arm, boils down a lot to a relationship that the investigator has with a given patient. So if the patient is convinced, for instance, that you were doing this not purely for an academic reason, but also because you have the patient's best interest in mind, that kind of patient-centric thinking is a really important place to start, really probably for any clinical trial. I think it's especially true in rare disease. And Dr. Blody mentioned the coordinating center. I couldn't agree more. Having really good clinical coordinators who know the patients, who can engage the patients, who can follow patients is extremely important. That's kind of your face to the patient community. For rare diseases, a lot of these diseases have patient support groups, patient advocacy groups. And so engaging with the leadership of those organizations at an early point is really helpful in my experience. And then just lastly, we live in a digital age that's filled with social media, and leveraging that social media, whether it's Twitter or Facebook or Instagram, can help you in your recruitment. So I'll give an example, and here it's not necessarily even something that I've done. My lab studies the pathogenesis, the genetics of a rare ocular condition called uveal coloboma. It's a congenital malformation of the eye. And patients come to us from all over the nation, in fact, all over the world. And when they're here at the NIH, I think they have, by and large, a pretty good experience. We really try to give them that patient-centric feeling for their good and for the good of the research. Many of those patients have posted on various Facebook groups. Was at the NIH, was part of this genetic study, and it went really great. And I encourage you, if you have any questions, moms and dads of kids with coloboma, come on out to the NIH. Here's what they have to offer. And I can kind of tell whenever somebody posts something to that. We're not posting it, it's just the parents that are posting this. But we see an uptick in our recruitment. And in fact, recruitment has not been an issue for this particular study. So you can only imagine that if you were able to deliberately utilize the tools of social media, how well you might be able to do under those circumstances.
Having information that, say, Dr. Brooks provides on a website for this support group really does help the patient and the family, and helps us too, because we are able to get information out to lots of people. And it's tough to get many questions because people will start to have lots of questions when they realize they have a rare disease or their child has a rare disease. But having sources of information that they can read about, I think helps us really stay focused on what we need to do and gives the patient that knowledge and understanding of where we are in the study and where we are in the knowledge base of this disease. I actually think it can help cut down on some of the questions that we have to field, because we want to just put it out there so people have access to it to begin with.
So Priya, one thing I might add to my previous comments is that we've talked throughout this webcast about the importance of natural history studies. So obviously, if you have established a natural history study, you already have a cohort of patients that you're following and have a relationship for. And so that would be an additional strategy, if you will, for recruiting patients when you're ready to have a clinical trial, to start thinking about your clinical trial even before you're ready to actually start your clinical trial, and be thinking about it in the course of your natural history study.
So how important is transparency and communication with patients and with their families?
I think transparency is extremely important. It's important for patients to understand the nature of the trial design, for example. Is there a chance that I might be on a placebo group? Are there potential risks to the treatment that you are offering for me? When will I know whether the results are going to help people like me, regardless of what group I'm going to be in? And especially based on at least your best understanding in, say, preclinical data or from early clinical trial data, what am I likely to expect, right? A patient who has hand motion vision, if your treatment is designed to slow the rate of degeneration, or even doesn't have to be hand motion, it could be 20/80. If your intervention is designed to slow or even stop the rate of degeneration, you're not going to make that patient 20/20 again by that intervention. And so setting the expectations, not providing, you want to always provide hope, but you do not want to provide false hope in dealing with patients. And so that kind of transparency, even though they're kind of difficult conversations, is extremely important, I think.
Yeah, I completely agree. I think that patients do want that honesty up front. And I'm sure Brian would agree, it's some of the patients with the rare diseases that are most committed to the research, no matter what. They know that this may never change, their vision or their life, but it can lead to further progress. And even I think many patients understand that even if the outcome of this trial is will show that this treatment does not work, we have learned something, we need to take a new direction. There's a lot to be learned, even when the outcome is not exactly what we want. So those patients, I think, are very understanding and generally very willing to participate.
Dr. Brooks, based on your experience conducting ophthalmic rare disease trials, what advice would you give clinicians who are designing studies and writing protocols?
First and foremost, you want to be thinking ahead. You want to be identifying patients, identifying physicians or practices that are following a lot of patients. And as Dr. Blody had kind of talked about, coming to consensus with other practices if you're doing a multi-center study, doing that early, because it does take time, is very important. You want to engage your patient advocacy group, as we discussed, engage social media organizations. That will help to drum up support, to help you in your recruitment. Start designing the trial in your head as you're collecting your natural history data. Be looking at data in real time so that you can decide whether this variable that I'm following in my natural history study, that looks promising, or this one, you know what, we're not getting usable data. You might even eliminate that from your natural history study and perhaps substitute it with something else that you think might be worth. So thinking what will the paper look like at the end of my clinical trial in terms of the structure of the trial, even while you're doing your natural history study. If you think you might want to do a trial in a given space, be prepared to create new outcome variables. So we did some work in albinism. You mentioned Luxturna and the low luminance maze test. In albinism, we had to create a new outcome variable focused on the amount of light shining through the iris, which is inversely proportional to the amount of melanin pigment. So we gave patients a small molecule called nitisinone and observed whether that affected their pigmentation in a small pilot trial. Be prepared to kind of think out of the box. You may not be able to use the normal go-tos that we would normally do. And I think, as Dr. Clemens and Dr. Bloody have both mentioned, you want to work with the FDA early. You want to start the discussions early, especially if you're considering what might be considered a non-traditional trial design, so that you don't get ready to start and get a nasty surprise that the FDA isn't buying your particular line of logic.
Dr. Bloody, do you have anything that you would like to give in terms of guidance or suggestions or advice to clinicians?
Sure. I think it can be daunting to think of an individual ophthalmologist starting a clinical trial, but it can be done. And I think ophthalmologists know a lot because they're seeing patients. And so good observers will see that there are certain needs and certain changes in their patients that may be worth researching on a larger scale. I really like the idea of repurposing drugs. And so we are going to all get more information on that when we have kind of access to big data. We will be able to see, are certain drugs helping or hurting patients, and that may be something that an ophthalmologist could easily try if there's a new drug that has some promise. I would, before you go into any trial, share your ideas with basic scientists who certainly know a lot about any type of drug or class of drug. And I would also share with other investigators your idea and your protocol. It really is sort of a community effort. It's hard to think that one person would be the one that would create the whole trial. There's so many aspects of a trial that need the assistance and the knowledge of others, whether they're ophthalmologists, statisticians, basic researchers. I mean, there's a whole host of them. I remember hearing Jean Bennett talk once, who's sort of the main basic scientist behind the Luxturna drug and had worked decades on this drug, getting this idea to clinical trials. She says in her talk, describing the journey, that the clinical trial portion was the most complex, the most challenging of the whole thing. I thought, you just came up with this amazing gene therapy, and now but the steps along the way are really lengthy, but I think so rewarding. And if you're in a clinical trial, I think you'll see how rewarding it really is. So that's it.
Thank you. Thank you, everyone. And I'd like to wrap up with asking each panelist to share a success story about your involvement in a rare ophthalmic disease study, or what inspires you to continue to participate in research around rare ophthalmic diseases. Dr. Clemens, let's start with you, and then we can go to Dr. Brooks, and finally, Dr. Bloady. Thank you.
Thank you, Supriya. Gladly. As Dr. Bloady mentioned MacTel. I was also a part of the MacTel and getting the structural outcome that was used, is currently being used in the phase 3 study, I think for me was very rewarding. The phase 1 study for the neuroprotectant that is currently under study started when we were just understanding the natural history. So we had an underlying natural history study that was ongoing. And at that time, we had about 6 or 7 years of follow-up on those patients. And what we were learning was that visual acuity was slowly progressing in those patients. Actually, we had conducted an analysis, and on average, those patients lost about 1 letter of visual acuity per year. So, as was noted, the outcome for the FDA is usually best corrected visual acuity, and it's usually 15 letters is the bar. So if we were to conduct a study, as you can imagine, in MACTEL using best corrected visual acuity, it would be a very long, daunting study. This outcome in the phase 1, which was just a safety outcome, and that was okay with the FDA. We also used it as in phase 2, again, to collect more information and again, stronger information with regards to the relationship of structure and function, and was able to provide that information to the FDA and also to the European regulators, EMA, to show that, yes, indeed, even what we showed from the natural history data, which was a strong correlation with structure and function, was even stronger with our clinical trial phase 2 information, relationship with structure and function. From that information, they did allow for our phase 3 study, which was the pivotal study. So for me, that was very rewarding, especially given that MACTEL is a disease where we have no treatment.
I think I've talked a little bit about some of my experiences, some of my successes. The polyLOMA protocol that we've been doing and engaging social media as a lesson, albinism, creating new outcome variables and engaging the FDA, and Stargardt, again, engaging the FDA and using each patient as his or her own control based upon carefully obtained natural history study. And I want to thank Dr. Clemens and all those that have been involved in the MACTEL-2 project, because the success of one group of people in a rare disease often can pay forward. So in this particular example, the fact that MACTEL-2 showed that there was a relationship between retinal structure using an imaging modality, OCT, and retinal function. In the end, the FDA cares about function. And so the fact that that correlation exists now enables me, as somebody working in Stargardt, to say, you know, my primary outcome variable is going to be a structural outcome, because look at that result from the MACTEL-2 phase 2 trial. So I think that there is a synergy often created with successes in rare disease clinical trials research. In terms of why I stay with it, one, I just find it very rewarding and very fun. And as Dr. Clemens pointed out, a lot of these conditions, if not nearly all of them, have no treatments. There's nothing that we as ophthalmologists have to offer patients. And so the fact that you can think of potential new treatments, try those, and hopefully do some good, to me, that's a really strong motivator.
Yeah, that's great. Those are great examples. So Priya, I'd like to start with or discuss an example of a disease where we're still in the early stages of describing this condition and understanding it. It's in the natural history stage, and that is reticular pseudodrusen, which is a mouthful, but it's an interesting story. It's a condition that was described decades ago. It's very subtle, drusen-like changes. The Kleins here at the University of Wisconsin were one of the first to describe it, seen in fundus photographs. It really wasn't until OCT and even high-resolution OCT that we're able to better identify these pseudodrusen. We don't really know what they are. They're deposits under the retina, not under the RPE. And it really takes multimodal imaging to identify. So we do know that with an autofluorescence with OCT, and infrared, we will get the best chance of identifying these new drusen-like structures. And they're important because they've been shown, they correlate with some worse outcome in terms of geographic atrophy. So these are older patients. AMD is not a rare disease. We thought this feature was pretty rare, but it turns out it's not so rare. So we're getting more information on how often we even see these atypical pseudodrusen. And now there is a natural history study underway. It is taking a lot of images. It does mean a fairly long visit for the patient, but with the help of EMIS, we are, I think, able to get really good information on patients with drusen, with and without those pseudodrusen, and try to understand what happens, what is the natural history. We have come up with a common language. I think that took a while. We've also come up with some imaging strategies. So it's evolving, but it is an example of what we certainly thought was rare. Turns out not to be so rare, but it's been difficult to find. So we're really working on kind of uncovering the mystery behind these pseudodrusen.
And I would like to thank all our panelists for their incredible insights and perspectives on the challenging topic of ophthalmic rare disease clinical trials. We appreciate your time today. For our viewers, if you have any additional thoughts or questions, we encourage you to reach out to EMIS. You may contact me directly or others in the Ophthalmic Therapeutic and Research Unit shown here. We also hope you will join us for our other EMIS at Home events. Thank you for attending and have a lovely day.